Antisense compounds. Antisense compounds or the antisense therapy is another strategy in rational drug design. But here, in spite of the chemically synthesized compounds, we are taking into consideration genetics. So, a gene is majorly playing a role in antisense therapy or the antisense compounds. So, while the human genome sequencing has been happened, so during that time, the antisense therapy has come into vogue. If we see of the Watson-Crick model of DNA, deoxyribonucleic acid, it has certain base pairs which are called as ATGC, adenine, thymine, guanosine and cytosine, and they have a complementary strand TACG. They are double stranded and triple stranded, and this DNA Watson-Crick model which is developed is something like a double stranded helical shape where the nucleotides will be base pair with double bonding and triple bondings. So based on this, DNA gets converted to mRNA and then RNA. So these processes are known as transcription and translation. So antisense oligonucleotides have come from the DNA antisense strand. As we know, the DNA has two strands. One strand is called as a sense strand and the other strand is called as the antisense strand. Sense strand cannot be used in the protein synthesis by cell, whereas the antisense strand is the one which helps in the protein synthesis to the cell. So when it gets converted to mRNA, which is a single-stranded one, so to this antisense oligonucleotides are combined. Antisense oligonucleotides are the small nucleotides with the nucleotides number ranging from 13 to 25 or sometimes even 10 to 15 also. So these nucleotides are very important in gene silencing or knocking off the gene. For example, if we are having a problem in the human body and that is because of a defective gene, the person who is having diabetes is having a problem in the genetics, or else many of the diseases which are transmitted through the hereditary are also having some problems in the genes. If we are going to rectify that particular gene and the function, and if we are going to silence the gene or knock off the gene specifically, the next generation or the next level of this study will be a healthy gene or the healthy function has been remodeled. So here if we are taking a defective gene where we need the function to be appropriate from the genome sequence, in the genome from this DNA to mRNA conversion has happened and the antisense strand which has been developed from the antisense DNA counterpart will be binding with the mRNA. So because of the RNase H enzyme activity, this mRNA gets hydrolyzed when the antisense is bind. So then protein levels are decreased. As the hydrolyzing of mRNA happens, protein levels are decreased and the function is rectified. So in this way, the particular gene which is responsible for causing a particular disease, the sequence should be noted first. So once the sequence is known, then it is easy to merge the mRNA with the antisense strand, and then finally that particular defective gene is going to be remodeled.